we are talking about genome editing. Okay. Uh, I emphasized earlier too that genome editing has confused many minds. Okay. And we should be very clear about the capabilities of genome editing. Okay. It's not something new. CRISPR-Cas system is a new system, about 7 years old now. But genome editing is not that young. People started working with the zinc finger proteins first a few decades back, and then they moved to talons, and then this is the third step that they have moved to CRISPR-Cas system because it has some advantages over the other explored systems. What is the confusion? The confusion is whether this genome editing system should have the regulatory setups for them or not. And then in the beginning of this lecture on genome editing, I mentioned that it can be used in two ways. One is to repair the genome. It means you are not inserting anything additionally to the human genome, but you are just repairing the SNPs or the small DNA fragments which have got mutated, and the mutations are involved in some genetic diseases. Okay, so just repairing the nucleotide sequences, replacing one nucleotide with another one so that the genes can be functional is the application of this genome editing in DNA repair. Okay. On the other hand, the same system, due to homologous recombination and non-homologous end joining, can also be used for insertion of the foreign DNA fragments. And those DNA fragments could be the cassets, having a promoter, a gene, and a terminator of your choice, which is equivalent to GMO technology. So if you are going through that way, that you are incorporating gene cassettes, the diverse genes, then the genome editing technology is equivalent to GMO technology. So the biosafety regulations, they are equally applied there. But if you are doing just the DNA repair, you're not incorporating anything new. You are not silencing any human gene or the animal gene. Then you don't have to go through any regulatory affairs. Okay. So this is the biggest dispute among the people, and the confusion is generated that the technology is called genome editing. But if you just step down to the second stair, you will see it bifurcating into two different fields. And obviously, one of that requires biosafety regulations. Okay? So this must be very much clear. And last time, what we were talking about was actually the mechanism of this genome editing system. Right from the beginning, when it was identified to its current state. So we are just talking about at the moment about the current state, okay? The original basic foundation state, okay? And then later we will go to the current state of that. So I hope you remember this slide from last time, okay? And I mentioned about it, about the clustered repeats of the sequences which keep on accumulating in the bacterial genome whenever there is an infection by a virus, okay? And they are separated by palindromic repeats, which are shown in black here, okay? So this is actually an immune system of the bacterial cell, or you can tell that this is how the bacterial cells memorize that they were infected with a virus, and next time the virus infects, how would they combat that infection? Okay, so this is a memory system which is known as CRISPR locus. Okay, and this CRISPR locus is further supported by the enzyme which is involved in binding to the CRISPR and tracer RNA, and in a complex form, a complex of three components, they identify the foreign viral DNA and destroy it. Okay, so this was the basic theory behind it. And then we moved towards the next slide. This was the last slide that I discussed last time. So they are present on the bacterial genome like a single entity. Okay, within this single entity, there are three components: the CRISPR region, the tracer region, and the enzyme Cas9. This is, which is actually an endonuclease, which cuts the DNA internally. Okay. How they make a complex was shown here, that all of this CRISPR region is transcribed into a single RNA molecule. So that RNA molecule contains the repeats, which are shown in black, these repeats. Okay, they come here. And then the region between these repeats represents the viral sequences, the short viral sequences which are left in the genome. Okay, so alternating repeats separated by the viral sequences. So this is a single RNA molecule, single stranded and single. Okay, so what happens is that at each position where there is a viral sequence, okay, and a repeat of the palindromic sequence, the Cas enzyme comes and joins at that position. And at the same time, the tracer RNA also joins that complex. So there are three molecules: CRISPR, tracer, and the protein, the Cas9. Okay, but they are all joined together by the internal nucleotide sequences, and these are chopped down actually before the individual component becomes functional. Okay, so they are cut by certain RNases. Okay, and the RNases, they cut the RNA from different places and separate these entities so that they can be functional. And this part, if you see this light colored part, this is the virus DNA. Okay, single stranded virus DNA transferred to the messenger RNA. It means that it is single stranded and it will be complementary to the DNA strand of the virus. Okay, so if the infecting virus has the sequences complementary to this sequence of the virus which were present in the CRISPR RNA, they can complement each other. It means that this particle of three components can move around the DNA and find out if there is any sequence similarity between the CRISPR RNA protospacer. This is known as spacer or protospacer. Whenever you hear, in case of CRISPR-Cas system, a spacer or a protospacer, it means the same thing. Okay, it is always the remnant of a viral DNA in the host genome. Okay, so this is the part which is the protospacer or spacer. This will find its complementarity with the infecting virus. If it gets some complementarity, so what will happen? That this is a single strand of DNA, it will bind to the complementary strand of DNA, and the enzyme will immediately cut that region. So this viral DNA will be divided into two halves. If there are more regions here which are complementary to the same DNA, this component having another protospacer will bind somewhere else on the DNA and will make another cut there. So the incoming viral DNA or RNA will be cut at various places. When it is cut, it means it is lost. The fragments get separated from each other, okay, and they don't make a fully assembled viral genome. So it is the death of the infectious agent, okay. So this is how the system works. Is it clear?
Yes, sir.
Yes, sir.
Okay, any question about it? No? Then I will proceed further. Now this is the
सर एक क्वेश्चन है सर
जी
पिछली
जी जी
सर ये जो वो हो रही है कैश सिस्टम बन रहा है बीच में जो 3 है सर ये जो नजर नहीं आ रही है डायग्राम जिसके अंदर से एक ब्लैक कलर की वो इंटर हो रही है फिर आगे व्हाइट सा रीजन आ जाता है फिर ब्लैक फिर व्हाइट ये क्या है सिस्टम?
हाँ यही मैं आपको बता रहा था कि जो व्हाइट रीजन आपको नजर आ रहा है ना व्हाइट रीजन दिस रिप्रेजेंट्स ये cluster of the viral sequences which are the part of the memory. ठीक है? So
black color repeats.
These are the black color are repeats. Okay.
Okay.
So this is the DNA. This is the chromosomal DNA. आप किचन से बाहर आ जाएं यार. Okay. So this is the viral DNA sequences. These are the repeats, but these are all DNA molecules. The first thing is that this DNA molecule is transcribed into RNA, not messenger RNA, RNA. So you get a single-stranded RNA, and that RNA represents these repeats and the viral DNA. Repeat, viral DNA. Repeat, viral DNA. Repeat, viral DNA. Okay? So these junction of these, where it is cut, these repeats are actually, I will tell you, they are the tracer RNA molecules. Okay? They are complementary to tracer RNA molecules. I will show you later on. They are not actually the tracer RNA. Tracer RNA is separate, but tracer RNA actually goes and joins on this black region. Okay? So they are complementing tracer and these repeat sequences. I will show you later. And where they are cut that one repeat is always attached with one virus sequence, and the cut is always in between the protospacer and the repeat region. It means here. Okay, it will be cut. Next cut will be here. Another cut will be here. So this is how it goes on. So every different unit, these black and white, they are single unit. Okay. And the next one is again a single unit. So 1, 2, 3, 4, 5, 6 different entities of the 3 components will be formed. And each individual will be like this. Okay. One virus sequence complement, which is known as protospacer, and then the tracer and tracer bound to the repeated sequence. Okay, have some complementarity, and both these RNA molecules, they are attached with the Cas9 enzyme, making a 3-component complex. Okay, so I will move ahead And I will explain it to you in this slide. Okay. So if we look at CRISPR region, which are the cluster of the viral DNA sequences separated by repeats. When they are transcribed, what they make? They make a pre-CRISPR RNA. This is what is shown here. This is the pre-CRISPR RNA. Okay. So single-stranded RNA is formed, which is known as pre-CRISPR RNA. These are repeats, and then separated. The repeats are separated by spacers. What is a spacer? What is a spacer? A
स्पेसर सर वो वायरल डीएनए कॉम्प्लीमेंट्स हैं
Exactly.
जो मेमोरी स्पेस में
Exactly. Perfect. These are the viral DNAs, which are about 20 nucleotides long. Okay. Remember, the length of them is about 20 nucleotides. Okay. So once these pre-CRISPR RNA is made, Cas9 is separately expressed. Okay. The tracer RNA, which may be a 171 nucleotide long and later converted to 89 nucleotide by chopping of RNase 3. Okay. Here you can see all the three components joined together. All the three components joined together on this pre-CRISPR RNA. Here you can see them. Okay. What you see additionally, very, very clearly, that the tracer RNA, this one, which is produced here, okay, this RNA is now joining what? It is having complementarity to the repeats which are present here on the CRISPR region, the black ones. They are shown as single-stranded here. So the red one is complementing to the black portion, and the black portion is the repeats in CRISPR, and the red region is the tracer RNA. It means the tracer RNA is complementary to some extent to the repeats in CRISPR RNA. Is it clear?
Clear, sir.
Yes, sir.
Okay, that's good. Now, because the pre-CRISPR RNA is a single molecule, so all the remaining components like Cas9 and tracer RNA, they are binding at the specific positions on this pre-CRISPR RNA, but they need to be separated from each other because each of them is having its own specific spacer or protospacer. Okay, so they must be cleaved. The pre-CRISPR RNA must be cleaved to separate these entities, and this is done by RNase 3. It's an enzyme which cleaves here and it separates all the components here. But those components are not functional yet. They have to be processed by some unknown nucleases which chop down these hybrid RNAs and the protospacer in a very specific fashion, so that the final length of tracer RNA is about 75 nucleotides. Okay, this part, and then the CRISPR RNA, on the other hand, the length is about 66 nucleotides. At this point, you must remember, CRISPR RNA is a separate entity from the tracer RNA. So on this side, we are talking about tracer RNA, which is a separate entity. It's just bound by hydrogen bonding to the CRISPR RNA due to its complementarity of the repeat sequences. Okay, the rest protospacer is overhang, single-stranded RNA overhang. Okay, but it has got some of the sequences from the next repeat. So that must be chopped down so that the spacer should be now freely available as 20 nucleotide overhang. Okay, and then there should be no overhang on this side. Now, remember, this is a three-component system. The attachment of the tracer RNA to the CRISPR RNA is just by hydrogen bonding due to the complementarity. So two systems join together. And third system is the protein, which is a big protein, which is embedding all these RNA structures in it. This is very important. That's the reason I'm emphasizing it a lot. Okay, so this is how the three-component system assembles together to make an endonuclease, which cut the DNA from inside. Internal cuts, double-stranded DNA breaks are made by these complex. Okay? So this is what I will explain it later, that what important thing is that this complex of three components, it joins the DNA at a very specific site. But in order to identify the right place, it first identifies a PAM site. What is a PAM site? At this moment, just remember PAM, the word PAM. And the PAM site is on the foreign DNA, the virus DNA injected by a virus inside the bacterial cell. That is the double-stranded DNA which is coming from the virus. This three-component system identifies a PAM site. And then below the PAM site, it identifies that if there is any complementarity there to the protospacer. If there is complementarity, this double-stranded DNA opens up, okay, and the protospacer gets complementary to one of the strands, and then this enzyme, which is shown in blue color, it cuts the double-stranded DNA of the infectious agent, okay, so that the DNA is broken down into pieces and the infection is overcome by the host cell. Is it clear?
Clear, sir.
Okay, that's fine. So now, what is PAM site? PAM site is protospacer adjacent motif. This is protospacer. A protospacer is actually the virus sequence, which is 20 nucleotide long. And this protospacer is attached to the CRISPR RNA, the black repeat sequences. So the length of the CRISPR RNA is protospacer length, which is 20 nucleotide, plus the length of the repeat sequence, which is adjacent to that. And you remember, this is one entity. The other part is the second entity, which is known as tracer RNA. I said that tracer RNA has some complementarity to the repeated sequences within the CRISPR region. Okay, and you can see that now. These are the complementary sequences. Only a few nucleotides are not complementary, but most of the sequence of the tracer RNA at the 5' end of tracer RNA is complementary to the 3' end of CRISPR RNA. This is very important. Okay, so you must be very clear. The 3' end of CRISPR RNA is having some complementarity to the 5' end of the tracer RNA. If you look at the double-stranded DNA, at one end there is 3' end, at the other end there is 5'. On one strand it is 3', on the other strand it is 5'. So they are just getting double-stranded here. But if you heat them a little, say at 80, 90 or 100 degrees centigrade, you bring this molecule, what will happen? If you heat this complex of CRISPR RNA and tracer RNA, which are having complementarity and they are joined by hydrogen bonds, if you heat this in water, what will happen? Very bad.
Sir, repeat करें sir.
If you have a CRISPR RNA like this and the tracer RNA like this, bring them together in a buffer or water, okay? So they will make a hydrogen bond here and they will join with each other. Now, what if after the joining you heat that tube to 90 degrees centigrade?
Sir, वो de-annealing हो जाएगी.
Yes, perfect. The two strands will separate from each other. 90 degrees centigrade will denature the double-stranded DNA into single strands again. Okay? So this is the two-component system. If we keep the Cas aside, okay? If we just consider the nucleic acids within the CRISPR-Cas system, there is one nucleic acid which is known as CRISPR RNA. The other nucleic acid is known as tracer RNA. They are complementary to each other. They can bind to each other. But if you heat them, or if there are helicases there, they can open up these bonds very easily, and the two-component system can get separated. And then in the beginning, I also mentioned, are you hearing me well?
Yes, sir.
Okay. In the beginning, I mentioned this is a three-component system. Lower is the number of components, easier it is to handle them. Okay. Is there any mechanism that we can make the three-component system into a two-component system? And that's what they did actually in the laboratory. So from the three-component natural system, they developed a two-component natural system. Two component out of three. By what did they do? They simply made a loop here and joined that loop with this one. Okay, and I will show you later on. So this end 3' end of the CRISPR RNA was joined to the 5' end of tracer RNA so that the CRISPR RNA and tracer RNA can become a single entity. Okay, so I will show you how they did it. Okay, but they joined it. So after joining, they got a two-component system. Very easy. You make the CRISPR and tracer in one unit RNA, and then you make a protein that is Cas9 in a second component, and they can join together to do the same function of double-stranded DNA cutting. And that's what is used now in the laboratories. That is the CRISPR-Cas system, two-component system that we are using in the laboratories to do genome modifications. Okay, so now do you know what is PAM? PAM is promoter adjacent motif. Where protospacer adjacent motif, where is it located? Is it located on the gRNA, the natural gRNA? The natural gRNA is the protospacer plus CRISPR RNA plus tracer RNA. Okay, this is the gRNA. It is not there on the gRNA. Remember that. The PAM site is always on the target chromosomal DNA. Okay? The PAM site, remember, it is always on the genomic DNA of the infectious agent. Okay? So this protospacer finds identity or complementarity on the infecting virus. Okay? If this region has some complementarity to the infectant, only then it will join. But it also requires a PAM site on the infecting DNA, the target DNA. Okay? If the target DNA does not have the PAM site, this gRNA will not work there. Okay? Whether it will find the complementarity or not. Even if there is complementarity, it will not cut that DNA until and unless there is a PAM site adjacent to the protospacer. That's reason it's called PAM, protospacer adjacent motif. This is protospacer. The adjacent motif means just above this sequence, there must be a PAM site on the genomic DNA. On the gDNA.
सर PAM एक सिग्नल कह सकते हैं, PAM को एक सिग्नल.
It's not single, it's a nucleotide sequence, GNN. Okay, so I will show you, you will be very much clear shortly. Just remember that this gRNA requires first the identification of a PAM site. PAM site is a small nucleotide sequence, and then below the PAM site, it should have complementary sequence on the target sequence. Two essential requirements so that the gRNA can work on the infectious DNA to cut it down. So, you know what is CRISPR-RNA? By now, you are very much clear. You know about the tracer RNA. Okay? You are very clear. You can see the CRISPR-RNA on the top, the tracer RNA here, and I just mentioned PAM site. And I will explain what PAM site shortly. Okay? So, what I mean is, you must be now very much clear about these three sequences. Okay? Now, I'm going to the next slide. I said in this slide that the 3' end is not joined to 5'. If we heat this molecule to 90 degrees, the CRISPR RNA will separate from the tracer RNA. Okay? So Briner was one of the scientists who in 2014, he made very different types of loops, 6 different types of loops, okay, and joined the CRISPR-Cas region, this end, with those different types of loops, okay. So one end of the loop, the second end of the loop, okay. One end joined here, the other here. So you have a loop region here, okay. And that loops are represented like this, okay. So you have the tracer RNA, you have the CRISPR RNA, and then you get it joined by a loop, okay. Now, if you join by a loop, it can actually modify the activity of CRISPR-Cas system. So what he did was he designed 6 different loops, attached them with a CRISPR-Cas double-stranded form, that is at the complementary region, which I have shown here at this end, okay? And then he studied the expression levels of those RNAs with different loops, okay? And he identified that this loop, GACGGAA, this one, if attached to this end, does not have any repression of the transcription of CRISPR and tracer RNA. So it retains its production capability. But now what you can do is, I will go back to an earlier slide. Okay, here you see tracer RNA is located at a different location. CRISPR RNA is located at a different location, and both of them, they are separated by protein systems. Okay, now by doing this, what they did was they put the tracer RNA here along with the CRISPR RNA. They brought them to separated components together, joined them through that loop region, so that that RNA could be formed, that instead that we get a pre-CRISPR RNA, we get a pre-CRISPR RNA attached to a tracer RNA. Okay, so all the unit, all together, all this part, starting from here, going down to there, they are produced as one entity. The other entity is now the enzyme Cas9. It means that on a plasmid DNA, you can have two expression systems. One for this complex of CRISPR RNA and tracer RNA together to make a single-stranded RNA molecule, and then a Cas protein, just two components. And when they are transcribed, the enzyme is produced from the messenger RNA, the tracer RNA, it folds back to itself and attaches to the enzyme to make a functional CRISPR-Cas system. Okay, so this is what they did, that they simplified this just by adding this loop, and if you can see this region on the top. So now this is the basic structure of CRISPR-Cas. Okay, it is covered by the top panel. So on the next slide, I hope I have represented it again. Yes, now you can see the complete single component system. This on your right. What is on your right? On your right, you have tracer RNA. The spacer is always joined to the CRISPR repeat. Okay, and here you can see the 20 nucleotide protospacer, also known as spacer, is joined to the CRISPR RNA. Because these two components, the CRISPR RNA it's a repeat, okay, joined to the viral DNA, right? If you remember from the beginning. The viral DNA, the repeat sequence, it makes CRISPR RNA. This is a loop, which is a linker loop that they joined. They put in there to join the tracer RNA. This is the tracer RNA. Okay, it is joined to the CRISPR RNA, and CRISPR RNA on the other hand get a spacer. And this is the part, only the spacer region, that will join to the infectious DNA. Okay, and the enzyme which is around these molecules, it will make a cut if the spacer binds to its complementary region to the foreign DNA. But the essential thing is that the foreign DNA must have a PAM site. So I will explain now the PAM site. A PAM site, if you come here, what are these blue ribbons? These are the double-stranded DNA, the target DNA, the DNA of the infectious agent. No, now this line drawing is for the Cas enzyme, okay, and the two entities now, which are now a single entity. That this is tracer part, this is CRISPR part, and the CRISPR part has got the spacer region, okay. When they join the double-stranded DNA due to complementarity of the spacer region. This cut will only be made if just upstream to the spacer region on the chromosomal DNA, there is a triplet or 3 nucleotide sequence, which is NGG. It means that if there is an NGG on the DNA, on the target DNA, only then the Cas will make a cut 3 nucleotides away from the PAM site. It means 1, 2, 3 nucleotides, and the cut will be here. Okay, and this is the PAM site, NGG. This is on one strand, so it will be actually, this is represented here, but it should be actually here, NGG. NGG means after this N, 3 nucleotides away on the protospacer, there will be a cut. There will be no cut if there is no NGG here. So PAM site is very important for cutting of the double-stranded DNA with the Cas9 protein. That is the nuclease, endonuclease, because it is cutting the DNA internally, not from the edges. Not from the edges. It's not an exonuclease. It's an endonuclease, which cuts the DNA 3 nucleotides away on the 3' end of the spacer. This is the 5' end. You can see the 5' end of the spacer, 3' end of the spacer. So on the 3' end of the spacer, 3 nucleotides inside, there will be a cut. Is it clear or not?
Clear, sir.
Okay, that's good. So I am now very much sure that you clearly understand the conversion of a 3-component system into 2-component system, and then how does this 2-component system works on the infectious DNA or the target DNA. Okay, and the enzyme makes the cut only if it recognizes the PAM site on the target sequence. PAM site is never ever a part of gRNA. Remember that. PAM site only exists on the target DNA, not on the gRNA. Okay, so it's on the infectious DNA. This one, the blue part, at this position after the complementary region of the protospacer, there must be a PAM site. And that's important. Whenever you design a gRNA molecule in the laboratory, you identify first that does that target DNA has a PAM site, NGG on it. So you can design a protospacer only adjacent to the PAM site, nowhere else. So we will do a practical on that, don't worry. We will see in the coming slides that how we make a gRNA. Okay, don't worry about that. But remember, PAM site on the target sequence is essential to make a gRNA, to design a gRNA. Okay. Now look at this. This was not at the nucleotide level. Okay, but at the nucleotide level, you see this is the spacer, the protospacer, which is 20 nucleotides attached to this is what is this black colored sequence? Remember, this black colored sequence, which is double-stranded, is the target DNA, infectious DNA. What is this blue color region? A blue color region is shown as 20 base pair nucleotide sequence, and that is spacer or protospacer. What is the red region? This red region, now you can see the loop actually. GAAAA. Okay, this is a loop to join the CRISPR RNA. I will say CRISPR RNA because you see the viral DNA sequence and the repeat sequence. This repeat sequence is complementary to the CRISPR RNA. Okay, so this is the CRISPR RNA now, which has different loops on it. Okay, so at the nucleotide level, now you know that this part, this is the target, and this is the two component joined together to make a single component through this GAAA loop. Okay, so the protospacer has complementarity to the target sequence, but it will not cleave until it identifies that yes, the target sequence has got NGG just upstream to the 3' end of the spacer. This is the 3' end of the spacer, 3' end. This is the 5' end of the spacer. So, on the chromosomal DNA, the target DNA, there must be the sequence NGG. This can be a T, this can be an A, this can be a C, or this can be a G. N means any of the 4 nucleotides, followed by 2 Gs. Okay, this is the PAM site, and the spacer is now complementary to the rest of the region. It means that this target sequence will be definitely cut by the Cas protein, which is shown in yellow here. Okay, I hope the things are very much clear to you. And the arrows indicate that 1, 2, and 3. This is the place, A and G. The Cas will cut between A and G, 3 nucleotides away from the PAM site. DGG is the PAM site, 3 nucleotides at the 3' end of protospacer. 1, 2, 3, and there is the cut here. And this cut is on double-stranded DNA on both sides, which are opposite to each other. 1 cut here, 1 cut here. Double-stranded DNA chopped out, producing a blunt end molecules. Am I right?
Right.
Perfect. It means that you are understanding the thing. That the CRISPR-Cas system will cut the target DNA in the blunt end manner. No overhang at all. Okay, this is the natural system. Okay, we can use the natural system and we can do some modifications according to our requirements. And that is included in the modified CRISPR-Cas systems, which is the second part of this lecture. Okay, we are going through the first part at the moment. Okay, so CRISPR-Cas makes a double-stranded DNA cut in a blunt-ended fashion. Okay, producing the blunt end overhangs. The natural system does not care what happens later, but we want to use that cut for putting in a DNA fragment or religating that blunt-ended cut, either through homologous recombination or non-homologous end joining system. That is the application by the scientists. The nature works till the cut only, so that the foreign DNA should be destroyed. Okay, so I am moving ahead from here. One thing which I want to clarify, sometimes you may see the word gRNA, that is guide RNA. Okay? sometimes you see the word sgRNA. What is the difference between gRNA and sgRNA? gRNA means guide RNA. Sg means single guide RNA. No difference. You can write sgRNA, you can write gRNA. It just is confusing when people do it. Some people prefer to write it as sgRNA. Some people, they prefer to write gRNA simply. There's no difference in these words. So never get confused with that. Single guide, it's always a single guide RNA. You know, there's only a single 20 nucleotide sequence of the virus genome. So it's a single guide RNA. So you can say it's a guide RNA or single guide RNA. Doesn't matter. I hope this is clear as well. So you will not be confused by this anymore. Now, another important thing. You know what is protospacer? Can you tell what is the protospacer here? Can you read the sequence of protospacer from 5' to 3' end on this slide?
Yes, sir.
Somebody, नहीं यार कोई और बोले, सारी जवाब आप देते हैं, बाकी लड़कियां सोई हुई हैं. Read the protospacer sequence in 5' to 3' direction, please.
Sir, AATGG.
No,
G,
no, no, no,
G.
no, 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 no.
Sir, it's
Yes,
GTCACCTCCAATGACTAGG.
yes, you are right. Can I hear from somebody else? It looks like everybody's gone.
Sir, blue region, this is the protospacer region. Yes,
All
sir.
right.
GTCACCTCCAAT.
Okay, perfect. Excellent. So don't sleep, okay? Just come up quickly. So this is the protospacer region. That is the single guide RNA sequence, okay? Or guide RNA sequence. Where would it guide? It will guide to the target DNA. That's why it is called guide RNA or single guide RNA. Okay, and this is what actually you synthesize in your laboratory. Okay, so if you have a plasmid which has already got this sequence on it, and then it provides you the provision that you can add a 20 nucleotide sequence here, or you can actually get all this nucleotide sequence synthesized in a single go, and you have the restriction site here, you have a restriction site here at this end. You can simply clone a guide RNA after designing into a vector. That vector will contain a promoter, obviously. Okay. Last time I told you about the zinc finger proteins, designing of zinc finger protein. Okay. It is just cloned into the restriction sites. The vector already contains a promoter for that. It always contains the other regulatory sequence for that. Similarly, instead of designing a protein which is 400 amino acids in case of zinc fingers, you are just synthesizing a small nucleotide sequence which is a composite of the spacer plus the CRISPR RNA plus the tracer RNA. On the terminal ends, you have the restriction sites, and you cut that synthetic DNA fragment from the two ends and clone that DNA fragment into the vector, which has got all the other things, the promoter, which I will show you later. Okay, so I'm not telling everything simultaneously so that you can absorb it clearly. So you can clone this very small DNA fragment very easily in the plasmid and you are ready for transformation. That is so simple. It's a very, very simple technology, I'm telling you. Very, very simple. Everybody can do it. It's such simple. But I don't know why people say this is a very complex thing. This is not really. Not at all. So I will just move ahead and we will talk that at the point where we will design the GRN. That how do we design it? Okay. So that is the last part, but we are in the first part yet. Now, the specificity of the protospacer, what does it mean? Specificity means how much complementary this protospacer is towards the target sequence. What do you see? What is the specificity here in this region? Can you tell me how much is the specificity? बोले ना. It's 100%.
Sir,
It's 100%. The complementarity is 100%. So the specificity is 100%. You see, C is complementary to G, A is complementary to T, G is complementary. This is target sequence. This is your space sequence. They are 100% complement. If they are 100% complement, this means they are 100% specific. If you don't see a complementarity here or here or here, you will say they are not 100% complementary to each other. So the specificity is not 100%, but lower than that. Okay? Now, if we look in those regards, that how much should be the complementarity of the protospacer with the target sequence, this is what I'm going to talk now, specificity of protospacer. And this slide is very important in telling you that. If you look at the top, what is that? This is 20 nucleotide long protospacer. Okay? 5' end of the protospacer, 3' end of the protospacer. All this red line covers the protospacer. And what is this GGG? Tell me what is this sequence? What is
PAM.
that? PAM site, exactly. And this is, where is this located? On the gRNA or the target DNA?
Target DNA.
Yes. Whenever you see a PAM site, it means it's a target DNA. So it means that what this slide means, if you understand, this is PAM site, this is the protospacer sequence, okay? It means that what are these lines which are going down and going up? This line tells the yellow here is a G, okay? And G is going towards the positive side. If a protospacer has got a G here at this position, it will be a preferred sequence. The protospacer will efficiently bind to the target sequence. That is the preferences of the nucleotides in the protospacer. That protospacer is the heart of gRNA. Okay? So you need to select a DNA sequence on your target sequence for cutting purposes. Looking at these preferences, that this is what position is that. Okay? This is NGG. It means this is the PAM site. So on the PAM site, at the position number 20, it means here, this T, here, on the protospacer, that is at position 20. This is position 1. This is position 20. At position 20, the favored nucleotide is either a G or an A. A is red, a G is red. So you take a sequence for the protospacer which has got either a G or an A at the 20th position of nucleotide. And if you have a C or a T, C is green, T is blue, it is a disfavored. Your gRNA is inefficient. gRNA means the protospacer sequence will be very inefficient in binding to the target DNA. If you want that your protospacer should efficiently bind to the target sequence, it must have a G or an A at the 20th position. Now, when I have started talking about this, I should better come to two things which are very important. Here I have written it as seed region, which is 12 nucleotides long. And this starts from the 3' end of the protospacer to this position. Okay, 8th position. So 12 nucleotides are the seed region which determine the binding efficiency of your protospacer with a target DNA. That's only 12 nucleotides out of this 20. Okay, the rest of the 8 nucleotides, they don't have very high preferences for any of the nucleotides. They should just be complementary. Okay, so you just need to concentrate at only a few positions. Position number 16 and position number 20. That at position 20 on a protospacer, the nucleotide sequence should be either an A or G, and at position number 16, it should be a C there. That is highly preferred for most efficient binding of the protospacer to the target sequence. I'm sure you are clear. So you don't need to consult actually this slide. I just wanted to tell you that these are the preferences of the nucleotides on the protospacer for efficient binding of protospacer to the target sequence. So once you know it, you make it a rule. You can write it in a single sentence that there should be an A or G and the protospacer at the 20th position. This is one rule. The other rule is there should be a C at position 16. So when you are selecting a protospacer on the genomic DNA sequence, you prefer to go to those regions which have a C at 16 position and A or G at 20th position. So you can, there are many more, you will see a lot of choices of selecting the protospacer. So you select the protospacer based on these two rules. That's simple. Okay, and we will see that in coming slides. Don't worry about that. But I hope you understand that position 20 and position 16 is very important for the appropriate selection of a protospacer from the genomic DNA, where you want to target the gene for cutting that DNA by the Cas protein. Okay? Is it clear? Any question?
No, sir.
Okay, good. Now, these are the rules that we make from these studies. Rule number 1. The 3' end of the DNA target sequence must have a protospacer adjacent motif. That is the PAM sequence. This one, blue region, GGG, this NGG. Okay, that this is one rule that The protospacer 3' end must have an NGG on the chromosomal DNA, not on the GRN. Remember that. The 20 nucleotide upstream of the PAM sequence would be the targeting sequence. So if you have an NGG, you select 20 nucleotides upstream to the NGG, that is your protospacer. That is one rule. Because the Cas9 nuclease will only cut if there is a PAM site at the 3' end of the spacer, the protospacer. Now look at this. I have taken the sequence. You can analyze any sequence, which you will see 2 Gs are present repeatedly, some nucleotides apart. So finding a PAM site is not a problem at all. If you have sequence on the Word file, just write GG in search menu, and you will see all your sequence will be having a lot of GG. And just one nucleotide above the GG will make it NGG, okay? And that will be the PAM site. So this will act as PAM site. This sequence will again act as PAM site. So this sequence will again act as PAM site. It means that you have no problem at all in identifying the target sequences on the genomic DNA, the target sequence. If you find a PAM site, you can take 20 nucleotides from here, and this is your gRNA. Any difficulty? Any problem? No problem at all.
नो सर,
Just
लेकिन
put
सर
the-
रिपीट्स किधर होंगी?
This-
इस मैप में रिपीट्स किधर होंगी सर?
You don't have to look for the repeats at all. You just take that sequence from here. All that sequence from here. Put the spacer that you have selected here. आपने जो स्पेसर यहाँ सेलेक्ट किया है, इस जगह पे, वहां पे आप AGG ना लगाएं, वहां पे वो सारा ट्रेसर क्रिस्पर और ट्रेसर RNA का सीक्वेंस लगा दें. Because when you get it synthesized, it's not the single protospacer 20 nucleotide region that you get synthesized. You get synthesized these 20 nucleotides along with the repeat, जो कि CRISPR RNA कहते हैं जिसे आप. What is that CRISPR RNA? It's a repeat. Then repeat sequence followed by loop sequence followed by the tracer RNA. And that will be about 200 nucleotides that will get synthesized. Instead of 200 nucleotides. On this end, you put a restriction site. On the other end, at the end of tracer RNA, you put a restriction site. According to the vector sequence. The vector no sites मौजूद है कि नहीं. You select the sites from your vector, just downstream to the promoter region, where you are going to do the cloning. The synthetic DNA that you have got synthesized, that is double-stranded DNA. Okay? You can cut with those restriction lines, and you can ligate that digested fragment into the downstream to the promoter, into the vector, which is the gRNA vector. Okay? That's so simple.